Hello, my name is Renee Schmidt, and this is my partner, Sam Gillespie, and today we are going to be talking about Candida albicans. So Candida albicans are a yeast, which is a type of fungus. They are commensal, meaning they are part of the human microbiome and normal flora. They are also an opportunistic pathogen, meaning when given the chance, they can cause infection in humans. So under normal conditions, they are harmless to us. But in, say, immunocompromised patients or in patients with broken skin, they can cause things like infection. So for example, they can cause oral thrush in infants, yeast infections in females, and in immunocompromised patients, they can cause things like sepsis, which is blood poisoning. And because of this, and the fact that yeast cells are eukaryotic like we are, sepsis is hard to treat. Because these antifungals not only kill the yeast cells, but they also kill our cells. So too much of it can kill us. So it's kind of like cancer. You can't kill the bad cells without killing the good cells. And this is important to diagnose early so that it can be treated early, or so it doesn't get to the point where treatment can also hurt us. So Candida albicans are diploid. They are similar to humans. They have 2 copies of each of their 8 chromosomes. They reproduce by asexual reproduction, meaning that the parent produces an offspring that is genetically identical to itself without the fusion of gametes. And because of this, there is less genetic variability. So they essentially create a clone of themselves. And to this day, there has been no evidence that they can undergo sexual reproduction like some other yeasts can. So we were studying loss of heterozygosity, or I'm just going to call it LOH for short. So why is this important? It's important because loss of heterozygosity in Candida albicans can make them resistant to drugs that are meant to help us. So for example, in the mutations in MRR1 transcription factors can become homozygous through LOH events, resulting in a dramatic increase in drug resistance. In humans, there's also evidence that LOH may play an important role in the development of cancer through inactivation of tumor suppressor genes. So this figure details the different clades of Candida albicans. This figure was taken from Dr. Pujol in Seoul. It talks about DNA fingerprinting of different strains that are classified into these clades. So we have clades 1, clades 2, clades 3, and South African clade, European clade. So what a clade is it is essentially a family within a species, meaning they all originated from a common ancestor. So as you can see, clades 1, 2, and 3 are more prevalent in North America. The South Africa clade is more prevalent in South Africa, and the Europe clade is more prevalent in Europe. So for example, in the North America set, 136 strains were fingerprinted to create this dendrogram. And for this study, Dr. Pujol provided us with 3 strains from each clade, as well as the 1 from South Africa and the 2 from Europe. So we were studying loss of heterozygosity in single nucleotide polymorphisms, or SNPs. And what these are is a 1 nucleotide difference within the same region. So our DNA is made up of a sequence of letters, A, C, G, and T. These are nucleotides. And what SNP is a 1-letter difference in these regions. We use restriction enzymes, which are molecular scissors. We use restriction fragment link polymorphism. And we use PCR to amplify regions around the SNPs that contained a restriction enzyme site. We digested the amplified regions with the appropriate restriction enzymes to assess LOH. We amplified a piece of DNA, which is bound by forward and reverse primers, and the regions were cut in 2 with these restriction enzymes. So these are the different primer sets that we used. We used both the forward and reverse primers. And the restriction enzymes we used was ALU1 and TAC1. And as I said before, they were used to cut the amplified regions within the primers. So as you can see, first number, where's the pointer? There we go. First number under the SNP column represents the chromosome that the SNPs are located on. And with that, I will hand it over to Sam to talk about the methods we used.
Thank you, Renee. So to conduct this research, we first of all had to extract our DNA, which we used with some chemical extraction using enzymatic and chemical disruption, followed by some purification. We would then use this purified DNA and put it into PCR to then amplify specific DNA strands. We use the restriction enzymes to cut this DNA and then use gel electrophoresis to visualize the results. This is our very famous DNA quantification machine, also known as the nanodrop. The nanodrop uses electromagnetic waves in the form of light at 260 nanometers. The DNA will then be absorbed, and based off of the absorbance, it will be then given in nanograms per microliter. After we get a concentration, we will then dilute the sample to around 50 nanograms per microliter. That way, we have consistent amounts of data for our samples. For our PCR, we are typically going to run with 3 different stages. We first off need to boil the DNA so that the strands will then denature, meaning that we have double strands turning into single strands. Once the DNA has been boiled off pretty well, we will then lower the temperature to the annealing phase. This will allow the primers to attach to the single-strand DNA, which will then take up that space. Once we bump that temperature up to 72 degrees, we'll allow our polymerase, Taq polymerase, to run its course. The polymerase will then make a complementary strand to the single-stranded DNA. We will then repeat the cycle 30 times. So here's an example as to what I'm talking about in the PCR, and then I'll be using the laser pointer for the image here. The black dot here is representing the SNP alongside the region that we are looking to amplify. And then towards the middle of the image, you also see the holistic DNA with everything in it. After a couple of cycles, what we are looking to do is just to amplify that red region that also has that SNP with the restriction enzyme baked in. After about 30 cycles, we will have over 1 billion copies of this amplified DNA region. As for gel electrophoresis, I would like to acclimate everyone to the top of the screen and towards the middle, where you'll see these light blue rectangles. These are going to be the wells, or where we are going to load the DNA into the gel. Once we do that, we'll then apply a voltage and a current, and depending on the way of thinking, we are either going to be pushing the DNA or pulling the DNA through the gel. The main thing is that the larger the DNA fragments are, the higher up, more towards the wells that they will be, and the smaller DNA molecules, the lower, farther away from the wells that they will be in the gel. As for the restriction enzymes, these are known as molecular scissors, and for ALU1, it is a 4-base pair blunt cutter. Its sequence is ACGT, and the blunt aspect means that it will cut straight down the middle between the sequences. This is a little bit different compared to TAC1, which is also a 4-base pair cutter, but it also has a 5' overhang, which leads to this Z formation. Now, in the case of a restriction enzyme site with the SNP, an SNP is one change in a nucleotide in the sequence. And so I'm going to be we're going to be going between the last 2 slides. As I go between the last 2 slides, you can see that there is a change in the sequence. And since the SNP changes one of the nucleotides, the restriction enzyme site no longer exists. We are unable to cut this region from that single nucleotide polymorphism. So, in the case in which that this SNP is there and there is no cutting happening, this is what it will look like on the gel. You will have a singular, large, thick band that has no details next to it. This would also mean that it's homozygous for not having a restriction enzyme site for both chromosomes. If it is going to have this iteration with 1 large band and then 2 smaller bands possibly, this would be heterozygous, meaning that there is 1 copy of the restriction enzyme site between the 2 chromosomes. And then the last case is going to be these 2 smaller bands below what would be normally the undigested or the uncut larger band. This is indicative of having 2 restriction enzyme sites, 1 in each chromosome, also known as being homozygous for 2 restriction enzyme sites. So on to the data that we have with our results. For 4R96, this one enzyme is not going to be particularly good for our data specifically, mainly because if we look at clade 3, there's a little bit of overbanding here towards the top. And then as well, for clades 1 and 2, there is the possibility of there being some digestion, but we may need to look more into it to see if we can get some more concrete results on that one. For 4R95, this is a good indication of it being homozygous for no restriction enzyme sites. It looks very clean. How about how there's all horizontal with the bands? There's nothing with any special nature to it. Now, as for 1L23, for clades 1, there is a bit of some digestion here for clade 1, and as well here on the right side for clade 3. This is indicative of it being homozygous for 2 restriction enzyme sites. Now, for the clade 2, it is also homozygous for not having a restriction enzyme site for both the chromosomes. For 2R68 at the bottom, there is a possibility here for clade 1. However, for clades 2 and 3, it is still homozygous for not having a restriction enzyme site. For 3L76, this is a great example with our data. Clades 1 has a heterozygous digestion here, as well as a homozygous for 2 restriction enzyme sites in the second strain, and another heterozygous for the third strain within clade 1. If we go over to clade 3 on the right side again, we have another example of it being homozygous for 2 restriction enzyme sites. Clade 2 is going to have more of the homozygous nature with no restriction enzyme sites as well. For 5L167, clade 2 is going to be homozygous for 2 restriction enzyme sites, which we have not seen so far. However, clades 1 is a little bit inconclusive, but there's some potential. There's the possibility of there being a little bit of digestion here that may be a little bit more difficult to see. Clades 3 is pretty cut and dry. It is homozygous for not having a restriction enzyme site. And then here for our last 4, we're going to have the South African strains and the European strains. So for the South African strain, there is no restriction enzyme site for these 3 enzymes or restriction enzymes. And then for the Europeans, 4R96 and 1L23 do have some digestion in the European strains on the left side and the right side. As for 2R68, this actually has pretty good digestion between all of them. So the South African strain has a possibility of it being homozygous for having 2 restriction enzyme sites in both the South African and the European 1 strain. And then for the second European strain, it looks like it could be heterozygous for having 1 restriction enzyme site. Then for the negative controls, we have no DNA in those samples, so there is no evident bands listed there. So as for our results, to recap, these 3 SNPs may be more suitable for the future research, and the remaining SNPs at the bottom may need to have some further testing to see if they would be suitable for this type of research. Now, there are some primers that may be more useful in the future with this research. We do have hundreds of SNPs that do have these restriction enzyme sites baked into them, so we may need to look further into those to screen them. For some future directions, I will give an anecdote for a clinical aspect of this. So if a patient has a fungal infection within their bloodstream indicating sepsis, a phlebotomist such as myself would be poking this patient to get a sample of their blood with the fungus inside of it. These culture bottles are then going to be incubated for a period of around 3 days. The sample will then be inoculated and then plated, and this process would take around 3 to 4 days. And our research, which would go along kind of with what McKenna was talking about with the different hyphae formations in the different clades, we would like to possibly bypass this whole system. So depending on all these different restriction enzymes and if the future research would indicate with these enzymes that it may work, we would like to screen possible SNPs and then design primers that can identify these same SNPs directly. This would eliminate the whole restriction enzyme site protocol and also the incubation for the clinical aspect. So hypothetically, we would be cutting the time from 4 days-ish to around half a day. Some acknowledgements. We would, of course, like to thank our great and mighty leader, Dr. Enger, as well as Dr. Pujol for gifting us the primer sets, as well as the representative strains that we used within this research.